Give people a chance to join. All right, it's a minute past. I think if anybody else comes in, they can just miss the introduction. All right. Well, hello, everybody. Thank you for joining in on the Marine Institute Graduate Society Seminar Series. We meet every week on Wednesdays at noon. This is being recorded and will be uploaded to our YouTube channel, where you can find almost all of our seminars over the past couple of years. Before we start, I do want to provide a land acknowledgement. We respectfully acknowledge the territory in which we gather as the ancestral homelands of the Beothuk and the island of Newfoundland as the ancestral homelands of the Mi'kmaq and Beothuk. We would also like to recognize the Inuit of Nunatsiavut and Nunatuckavut and the Innu of Nitassinan and their ancestors as the original peoples of Labrador. We strive for respectful relationships with all the peoples of this province as we search for collective healing and true reconciliation and honor this beautiful land together. Now, please join me in welcoming DFO research scientist, Dr. Carrie Holt. Dr. Holt's studies primarily focus on salmon population dynamics and developing benchmarks for management decisions. We were talking about this before the seminar, but she's also on the Costiwick Marine Fisheries Subcommittee, which is funny because we just had Dr. Sarah Leonard speak, who's also on that committee. Dr. Holt graduated with her Ph.D. in 2007 from Simon Fraser University, and then joined the Punt Lab, excuse me if I said that wrong, Punt or Punt Lab at the University of Washington as a postdoc, where she incorporated satellite data on ocean conditions to help groundfish species recovery, which I thought was really neat. So, but with that, I'll let you take over.
Thank you very much, Colin. It's an honor to be here. So today I'm speaking from Nanaimo, British Columbia, which is the unceded traditional territory of the Nunamuk First Nations. I Kwanasi Lamnama, that means it's good to be here with you today in Hulkaminam, the language of the Nunamuk First Nations. So today I'm going to talk about sustainability of salmon fisheries, accounting for population structure and time-varying productivity and assessment and management. So this is where I am. So on the other coast of Canada, and Nanaimo, on this and Nunamuk, the coast, part of the Coast Salish First Nations. I would like to acknowledge my co-investigators in this work, Kendra Holt, Katerina War, Luke Workington, Catherine Mickelsons, and a technical working group of 29 members from DFO and First Nations. So first I'll give a brief outline. My talk will have two components to it. The first is on the population structure. The schematic on the left just shows some of the population structure for Pacific salmon in BC. So why does population structure matter for sustainability, and how should we assess and manage Pacific salmon to preserve population structure and component biodiversity? In particular, I'm going to talk about identifying limit reference points for Pacific salmon that account for that population structure. And the second part of the talk, I'll discuss some of our recent work on evidence for time-varying biological processes and population dynamics for salmon, and changes in reference points or biological benchmarks to account for temporal variability in productivity. So answering questions such as how does temporal variability in productivity impact the estimation of reference points? What are the consequences if it's ignored? And should these benchmarks or reference points account for temporal variability when used for assessment and management? Where this schematic shows some of the time-varying patterns in productivity that we've seen across stocks in Fraser sockeye. So first, why does population structure matter? Population structure can arise because of differences in adaptive traits among populations within a species. These traits can be life history traits, inherited behavioral adaptations, or other genetic markers that can result in differences in population status and trends. This results in diversity in population level responses to threats and population trajectories. Portfolio effects have been established for numerous salmon stocks defined as population aggregates that contain a population and life history diversity, which produce temporally stable recruitment. And where these portfolio effects exist, the aggregate population abundances tends to be more stable over time than in cases where these portfolio effects and the underlying stock structure do not exist. And so population and life history diversity can buffer species from environmental variability and contribute to long-term sustainability through the differing responses to variable conditions. And so I have this ribbon on the bottom showing the different components of the presentation so that you can kind of follow along with what part of the presentation I'm on. So this slide shows one of the important early papers showing evidence for the portfolio effect within Pacific salmon by Schindler et al. So they looked at 3 different levels of population diversity for sockeye salmon. The first was this Bristol Bay sockeye salmon aggregate, so the aggregate of all the populations in Bristol Bay. Then 9 component rivers within Bristol Bay. And then within one of those rivers, the Wood River, they looked at the streams within that river. So they have these 3 different levels. Then they looked at the interannual variability in population abundances, and they show that on this bottom plot here, that the interannual variability is much higher when you're looking at that stream level compared to when you're looking at the aggregates to the river or to Bristol Bay. So when you integrate and each of these streams or rivers, they have different they vary in their run timing. So when you integrate diversity in these run timings across these different streams and rivers, then the population abundances tend to be more stable. So you get the stability of the aggregate. And what's shown here in this bottom plot is when we have no age structure, and we're just looking at a single age maturity. However, salmon return at multiple ages. This picture just shows the size at at different ages at return. And when you incorporate the diversity in that the age of maturity, then we see that the interval variability, when you include that age structure, is much lower. So that also contributes to the portfolio effect here. And so we also see changes in this within species diversity over time. And so this is a good paper that demonstrates this by Price et al., showing an erosion of within species diversity for Skeena sockeye salmon. And so this top picture shows the gillnet fishery in the Skeena River in 1915. And this gillnet fishery is blamed for diminishing a component of the sockeye population that returned to spawn later and were typically larger, so that the smaller and earlier timed fish were able to escape the gillnets. So that led to some erosion of that within species diversity that was amplified by this enhancement that was on one component of on the main stem of the Skeena that supported a large commercial fishery. So this map here just shows the Skeena River, and which has numerous populations within it that can be aggregated into these 13 complexes that are shown along the border of the map. And these plots on the outside show the distribution of abundances over time, where the yellow and blue show the historic abundances and the gray are the recent abundances. So we can see that in most of these components, the abundances are becoming smaller. The red, however, shows where there's been enhancement. So we see that for a few of these stocks, there's large enhancement in recent years. And so what this means is that there's been a contraction in the spatial distribution over time. This is that same map here showing the change in distribution, where red is a minus, the dark red is minus 100% in the abundances, and dark blue is increasing 250%. So we see that there's a loss of these fish on the outside streams, and an increase in the main stem, which is where the enhancement is focused. So this has led to this deterioration of this within species diversity for this aggregate. So this raises the question, how should we assess and manage Pacific salmon to preserve this population structure and component diversity? So this is relevant for us at DFO. We have a revised Fisheries Act, which provides a legal mandate to implement measures to maintain and rebuild fish stocks. And so the text of this Fisheries Act says that in the management of fisheries, the minister shall implement measures to maintain major fish stocks at or above the level necessary to promote the sustainability of the stock, taking into account the biology of the fish and the environmental conditions affecting the fish. If the minister is of the opinion that it's not feasible or appropriate for cultural reasons or because of adverse socioeconomic impacts to implement the measures in subsection 1, the minister shall set a limit reference point and implement measures to maintain the fish stock above that point, taking into account the biology of the fish and the environmental conditions affecting the fish. So our role is to develop these limit reference points for Pacific salmon. Important to that text is that it pertains to major fish stocks. So a major stock for Pacific salmon is a group of one or more Pacific salmon populations or conservation units that are managed together with the objective of achieving a joint status. And so there are 69 of these stock management units, what we call SMUs, in BC and the Yukon for Pacific salmon. Also important for this text from the Fisheries Act is the definition of the limit reference point. So a limit reference point is the stock status below which serious harm is occurring to the stock. At this point, at this stock status level, there may also be resultant impacts to the ecosystem, associated species, and long-term loss of fishing opportunities. So importantly, the Fisheries Act requires that for each stock management unit, there is one limit reference point. Also relevant to identifying LRPs or limit reference points for Pacific salmon is Canada's wild salmon policy. And the goal of this is to restore and maintain healthy and diverse salmon populations and their habitats for the benefit and enjoyment of people of Canada in perpetuity. One of the objectives of this policy is to safeguard the genetic diversity of wild Pacific salmon by maintaining and protecting conservation units. And these are groups of wild salmon sufficiently isolated from other groups that, if extirpated, is very unlikely to recolonize within an acceptable time frame. And there are over 400 conservation units, over 5 Pacific salmon species in BC and the Yukon. And these are identified because these groups of salmon are exposed to unique freshwater and marine ecosystems that result in localized adaptations. They may have different life history types and different ages of maturity that are inherited, and different genetic markers. To identify these, authors looked at neutral genetic structure within dendrogram trees based on published data on microsatellite loci. So these conservation units are nested within the stock management units. So this is an example from coho salmon showing an inset of the southwestern BC, showing on the left here, there are 11 conservation units in this area, and this same area covers 3 stock management units. And these CUs are nested within the stock management unit. Within the wild salmon policy, biological benchmarks are identified that delineate 3 zones of status: green, amber, and red, which represent increasing conservation concern requiring increasing management intervention. So this lower benchmark between the red and the amber zone is identified at the level to ensure that there is a substantial buffer between it and any level of abundance that could lead to a CU being considered at risk of extinction by COSEWIC. Given the similar intent of the LRPs under the Fisheries Act, that is to avoid serious harm, in the lower benchmark on the wild salmon policy, that is to buffer against extinction risk, we've argued that it's practical to use these lower benchmarks to represent serious harm at the CU level, but we need to address this challenge of different spatial scales of application. So under the Fisheries Act, we have 69 stock management units, and under the wild salmon policy, we recognize that serious harm occurs when those conservation units, the 400 of them, when any one of them is depleted. So our challenge is to identify LRPs that integrate status across component CUs. So our goal was to develop these LRPs for salmon that account for the biodiversity of the component CUs. Our goal was to develop a toolkit to identify LRPs given variability in data types and quantities and qualities across all of those CUs. And this was work that was supported by that technical working group that I mentioned. So DFO's precautionary approach states that LRPs are usually in units of biomass, but other units can be considered, so including escapement or yield and effort control fisheries and other biological characteristics. So generally we think about it in terms of LRPs along a gradient in stock status and stock abundances or biomass. This is a schematic from DFO's precautionary approach framework showing that limit reference point between the critical and cautious zone along a gradient in stock status. However, we can also think about it along other metrics. And so we've proposed a CU status-based LRP where an LRP might be breached when one or more component CUs is below its lower biological benchmark or in the red zone. And so we could think of this as a proportional metric where you're looking at the proportion of those CUs that are above that red zone. So here is an example of this, of a CU status-based LRP, where the LRP could be 100% of CUs with status above the red zone or above the lower benchmark. Here's a hypothetical stock management unit with 5 conservation units, with 4 in the green zone, 1 in the red zone. So in this case, the SMU would be below the LRP of 100% of CUs. So we can also think about these aggregate abundance-based LRPs. So this slide shows a summary of methods. The CU status-based LRP is what we are recommending under the Fisheries Act because it links closely to what's required under the wild salmon policy, and we already have assessments methods developed at the conservation unit level. However, we can also consider aggregate abundance-based LRPs. And these may be used in addition or supplementary to these CU-based benchmarks, LRPs, when abundance-based reference points are required in the specific management context. And in our work, we've identified two types of aggregate abundance LRPs that I'll describe here. So to be aligned with the wild salmon policy objectives, these aggregate abundance LRPs need to be at an aggregate abundance associated with an acceptable probability of all component CUs being above red status. And so we've considered 2 methods, and both are based on the assumption of a predictable relationship between that SMU level abundance and the probability of all component CUs being above red status. So we consider these 2 types, a logistic regression-based LRP and a projection-based LRP. And they both use that same assumption, but the logistic regression model uses a statistical model fit to historical data to identify the LRP, whereas the projection LRP is based on projected population dynamics. So I'll describe this first one first, this logistic regression. So the way we do this is that we start with CU level spawner abundances relative to their lower benchmark. So this is a hypothetical stock management unit with 4 component conservation units. And this shows their time series of spawner abundances over time relative to a lower benchmark. We can then create an aggregate data set where for each year we have the aggregate abundance and then whether all of the CUs are above red status, yes or no, coded as 0 or 1. And then we can plot those and fit a logistic regression model to those data to identify the probability that all conservation units are above their lower benchmark along that the x-axis of aggregate abundances. We could then identify an LRP associated with, for example, a 50% probability that all conservation units are greater than their lower benchmark. Or we can look at other probabilities, such as a 95% probability of all conservation units being above their lower benchmark. So some of the challenges with this logistic regression approach to identifying LRPs is that these logistic regressions have poor fits for many stock management units or the models don't converge. There can be data limitations where stock management units that there are no years when all CUs have been greater than their lower benchmark, or the historical time series may not represent current or future population dynamics. So we considered an alternative to this that was a projection-based LRP. So to implement this, we developed a population dynamics projection model using a model for a simulation model for salmon that had already been developed and published that included a stock recruitment relationships by conservation unit with variability in recruitment residuals, variability in age of maturity, and covariance in recruitment residuals among CUs. And we applied a fixed exploitation rate with implementation uncertainty within this simulation. So we then projected the CU level abundances to an equilibrium state, shown here for this example with 5 conservation units. And then based on that, we can characterize the underlying relationship between the aggregate abundance and the probability that all CUs will be above their lower benchmark by aggregating that information from all those projections, all the Monte Carlo trials and years, and binning them to create this plot of aggregate spawner abundances and the probability of all CUs being above their lower benchmark. We can then, similarly to the logistic regression, identify an LRP associated with a variety of probabilities of all of the component CUs being above their lower benchmark. So, as I mentioned, these aggregate abundance-based LRPs are considered supplemental to that CU-based or proportional-based LRP. And then based on these different approaches, we've developed some, and our application to 3 case studies, we've developed guidelines for implementing these LRPs that are accounting for the CU level population structure. So we developed a stepwise procedure on how to implement them and accounting for data deficiencies in a sequential way. And this work and these guidelines, the methods and guidelines, have recently been through a CSAS review, the Canadian Science Advisory Secretariat review last week, and they've been accepted. So there'll be 2 papers, research documents that will come out of this work, one showing, describing the guidelines on implementing these LRPs, and a second on a case study application of these methods. Also a science advisory report will be published somewhere in the fall. So far I've talked about kind of the spatial or CU population structure for identifying and those issues around the population structure for identifying reference points. The second part of this presentation, I'm going to talk about temporal patterns and how we can account for the underlying changes in temporal biological processes when estimating reference points. So we have evidence for time-varying population processes associated with environmental changes. This is an example from Suswelski and Hollywood, where they pulled some examples from the RAM legacy database for a variety of species, where they associate changes in biological processes associated with recruitment and spawning biomass to different environmental conditions. So production or biological process can be altered by changes, or production can be altered by various changes in population processes. For example, changes in temperatures can cause changes in growth, changes in phenology can produce changes in recruitment, and changes in ocean current patterns can influence prey availability for larvae and juveniles and or settlement in nursery habitat. We've also seen declines in productivity for Pacific salmon in particular. So there have been some high-profile declines in productivity for salmon stocks across North America. For sockeye salmon in particular, this resulted in a federal inquiry into the declines of sockeye salmon in 2010, 2011, which is documented by the Cohen Commission. This is bottom panel here. We see large, for Pacific salmon, we do see large variation in productivity over time. This schematic here shows productivity changes for 19 stocks of Fraser River sockeye salmon, where the red are negative deviations, red circles, and the blue are positive deviations over time. So we can see kind of some variability early in the time series, and then around 2005 brood year, we see some consistent declines, and those are the declines that resulted in that federal inquiry, followed by some variability and then recent, much larger declines. So there's consistent evidence of declines for Fraser sockeye salmon. The methods to account for time varying parameters have also been developing. These papers have extended the standard stock recruitment models to account for annual changes in the underlying population parameters. So this bottom right plot here just shows an example of the stock recruitment relationship and how we can vary, for example, the productivity parameter to over years to capture the changes in the underlying processes over time. Then one question is, given these changes in productivity, which may in part be environmentally driven, what are the impacts on reference points and biological benchmarks? So reference points or harvest control rules can be revised to account for time varying processes, and failing to account for them may result in biased biomass and fishing mortality based reference points. However, the evaluation of these time varying reference points show that they can lead to small increases in yield, but also associated with higher risks of overfishing. So, and these benefits of changing the reference points may be much greater if shifts are thought to be permanent. This example here on this slide is an example that demonstrates the complexity from updating reference points. So this is an example described in Suszewski and Hollywood, where both for showing the numbers of blue king crab and red king crab around the Pribilof Islands. So both red and blue crab are harvested in the same fishery, where the blue crab is considered overfished. If the low population size of the blue king crab is the new normal based on environmental conditions, then it may be possible to reduce reference points. Then the red king crab would not be limited by the requirement to rebuild the blue crab to original abundances, which may be no longer attainable. However, if there is a possibility that the blue king crab could rebound, protecting it from harvest until it does may be prudent. So this example demonstrates this trade-off that will likely emerge in the future. And then really the recommendations from that work was that we need to understand the mechanisms resulting in the changes in abundances for the species. So what are the implications of this kind of time varying productivity for Pacific salmon? So I'm going to spend the last part of this presentation talking about our work on impact of time varying productivity on stock recruitment based parameters and biological reference points for salmon from 2020. So first, we documented evidence for time varying productivity from stock recruitment relationships for sockeye in the Fraser. So this is one example for Stalaco sockeye, where the darker dots are the more recent data. So we first fit standard Ricker models to the data, and then a time varying model where the productivity parameter or the slope at low abundances was allowed to change over time. And so we show the evolution of that productivity over time, where the darker curves are the most recent. So we're seeing this decline in productivity. And then we can also plot that over time, where we see that this this model has an explicit time varying parameter shown in that right plot. And this changes in productivity can account for environmental driven changes in survival, for example, related to deteriorating habitat in freshwater or the marine environment in recent years. So what does this mean for reference points? So we show that this impacts our estimate of the reference points based on these two modeling assumptions. So the red is the SMSY based on the standard Ricker model, and the blue is the average of the time varying models. So in this case, we see that the average of the SMSYs from the time varying models tends to be much higher than that assumed from the standard model. And this is because of the difference in the assumed density dependence, that there is evidence for this overcompensation in the standard model that we don't see in the time varying model. So there's an interplay here between how the model estimates productivity and capacity, but it's not clear which of these benchmark estimates better reflects the true SMSY. We can't identify through empirical analyses alone because both are based on kind of estimation. And this is just one example of Silico sockeye from the Fraser. We did look at this over 12 different stocks. But we then followed this with a more rigorous evaluation of these reference points using simulation evaluation, where we knew what the underlying reference point was, and then we can compare that true underlying reference point to the estimates from both of the models. So this brought us to our objectives. I'll just describe those on the right here. So in simulation, to evaluate the biases and reference points estimates under different scenarios of time varying productivity using standard and time varying assessment methods. And then, to answer the question, should reference points account for temporal variability when used in assessment and management, we provided some best practices when applying time varying models for reference point estimation. So to do this, we developed a simple simulation model for a hypothetical sockeye salmon population with sensitivity analysis that focused specifically on the Fraser River sockeye. So this simulation model had two components: an operating model that simulated the parts of the system that we cannot control, such as the stock recruitment or population parameters, population dynamics, and the fishery dynamics, including stochasticity, and then the management procedure, which is the part that we can control, the monitoring and assessment, so the assessment methods that we applied, and a harvest control rule. In the operating model, we considered a few different scenarios for productivity and changes in the underlying productivity. Here I'm going to, so a kind of a constant, a decline, or an increase in productivity over time. I'm going to focus on this middle one in this presentation. That's that decline in productivity, because that's really what we've seen for the Fraser sockeye and for many salmon stocks across the coast. In the management procedures section, we considered a variety of different exploitation scenarios: a constant low exploitation, constant high exploitation, and then we also considered this step decline in exploitation, because this is really what we've seen for Fraser sockeye, where there was a very high exploitation rate until the late 80s, early 90s, followed by a much lower, more precautionary harvest rate. So I'll spend some time explaining the results based on that third one as well. We then repeated the simulation over years and Monte Carlo trials, and over these different scenarios, the productivity and the exploitation rate scenarios. And then we evaluated our deviations between our estimate of the reference point and the true underlying reference point, the true SMSY. And our estimates were based on that standard model that I showed before, and then that model that explicitly included that time varying productivity. So these are some of our results. As I said, I'm just going to focus on this scenario where we had this decline in productivity over time, though I do show the three different harvest scenarios. So the constant low harvest, constant high harvest, and the step decline in harvest. And this plot shows the mean percent error from the true MSY. So, how biased are our estimates of MSY for both the standard model in red and the time varying model in blue. So overall, we see that time varying model is much less biased. This is averaged over the years, and that we do see large biases in the standard model. So I'm going to spend a few slides just describing why this bias is happening. But for those of you who are really interested in stock recruitment modeling, for those of you who aren't, just the bottom line is that the time varying models were much less biased than the standard models. So we can kind of piece out these results a little bit by looking at the individual stock recruitment relationships. This curve here shows what the true model, underlying true model and the operating model was with a true SMSY. This is the temporal mean. And then we can layer on top of that this scenario where we had this large bias here with high harvest rates. And so this red band here are the distribution of curves from that simulation. So we have large uncertainty here on this right side. Looks kind of like a megaphone. And this is because we've got lots of uncertainty in where that density dependence happens. So what is the capacity? This model can't really tell what that capacity is. And superimposed on here, we have an example of some of the data points. So we see that we have all of the abundances are at low spawners. All of the data are at low spawner abundances because of this high harvest rate. And we have a few kind of a high recruitment early in the time series when the productivity was high. And so this results in SMSY that is overestimated. In contrast, when we look at this scenario where the harvest rate declined over time, this is what the band of the distribution of curves looked like. So we get this much more stronger evidence for density dependence here. SMSY tends to be underestimated in this case. And this is an example of the data points, the X's here. So here, under the declining harvest rate, there are some poor recruitment events that happen at high spawner abundances late in the time series when productivity is low and harvest rates are low. And so this is what we see for Fraser sockeye salmon. This is exactly what we see. And this tends to pull the orange curve downwards and results in an underestimate of SMSY. So basically, we see these biases in the standard model that are an interplay between the declines of productivity and the exploitation rate time series, and that the time varying model, this blue, tends to be less biased. But it's also important to realize that in addition to changes to these biases in SMSY, there's also temporal changes in the underlying true SMSY. So on the bottom here, we see as productivity declines, the true SMSY also declines. This SMSY is derived directly from productivity. So the question is, do we want to manage to lower SMSY values? This may result in relatively optimistic estimates of status, possibly reduced harvest restrictions, and increased exploitation. So this choice will depend in part on the underlying drivers of productivity. If the decline in abundances is due to understood persistent and irreversible decline in habitat, then adopting a lower SMSY may be appropriate to achieve management objectives. However, if the shifts are not well documented and possibly reversible, such as associated with declines in survival, for example, related to decadal scale variability and oceanographic processes, then more spawners may be needed to produce the same number of recruits compared with the historically high productivity regimes. And a precautionary response would be to keep them constant or even raise reference points while productivity is low. So we do need to kind of consider that, how do we want to respond to these reductions in productivity? So more broadly, we recommend several best practices when considering revisions to benchmarks or reference points due to persistent shifts in productivity. First is to document evidence for the degree in changes in exploitation. Also document evidence for changes in productivity. Document underlying mechanisms that could be causing these changes in productivity. And demonstrate that the change in the state of the productivity will persist long enough in comparison to the management plan so that changing the reference points will indeed respond to management needs. Calculate stock recruitment estimates and benchmarks with and without time varying productivity and compare values. Estimate and communicate the implications of uncertainty in these reference points or benchmarks, and then support decisions for benchmarks reference points with simulation models that include management procedures with the assessment and application of those reference points. So in conclusion, overall, from that first part of the presentation, I demonstrated how we're developing methods to set limit reference points for Pacific salmon that account for the population structure within the stocks, population structure and biodiversity under Canada's revised Fisheries Act, and that reference points can be adapted to account for temporal productivity, and when ignored, reference points estimates can be biased depending on the historical exploitation patterns. Models with time varying patterns tend to provide less biased estimates of SMSY, but that there can be unintuitive impacts of changes in productivity on resulting status, so we recommend caution when applying these. And as part of our next steps, we're evaluating the impacts of changes in productivity and capacity. So we have evidence that not only is productivity at low spawner abundances changing, but we have this changes in the carrying capacity of the population due to changes in habitat. And developing guidelines on when and how these reference points should be adjusted based on simulation evaluations and empirical case studies. And this is a DFO project funded for both myself and Brendan Connors and others. We have 2 postdocs, Dan Greenberg and Katerina War, who are contributing to that work. And I'd like to say thank you. Hi, Sepka. That's in Halkomenan language of the Sunamuk people.
All right, thank you so much, Carrie. Can you hear me?
Yes.
Okay. Yeah, so we have plenty of time for questions. If anybody has any questions, just go ahead and just chime in. It's fine. Pierre, I think I saw your hand up.
Hi, Carrie. Nice talk.
Hi, Pierre. Nice to see you.
Yeah, we don't get much of a chance to do that these days. No comment. So there's a couple of thoughts that crossed my mind when you were presenting, and the structure within the SMUs struck me as something that's really important here. Have you considered that there's a possibility that the CUs that make up these SMUs might actually be have different vulnerabilities to the harvest, and what kind of situation that would create in terms of identifying what a reasonable limit reference point would be? And then I've got a kind of little follow-up question to that.
Yeah, great question. Yes, in many cases, they are differentially vulnerable to harvest, and that they tend to, in those cases, they tend to not, the abundances don't co-vary very tightly. And so, one option is just to manage them separately. So to look at them separately, develop harvest control rules that have like time area based closures, or that are kind of at a finer scale. However, if there is a requirement for an aggregate LRP, because in some cases there's international management, like Pacific Salmon Treaty requirements, that there is an aggregate level escapement goal or aggregate level. In those cases, we do need to account for the fact that that the abundances don't co-vary very closely, and that there may be differential vulnerability, and so to fishing or to other threats. And so in those cases, these other methods, like this logistic regression method or this projection-based methods that explicitly account for that, how they co-vary over time, they tend to, if they don't go very well, they tend to provide LRPs that are quite high, because there's a good chance that at low to moderate abundances that at least one of those individuals will be below its lower benchmark. And so they tend to push the LRP quite high in those cases. So that's why we've kind of proposed these other methods, because they explicitly account for what the covariance is. Now the challenge is the methods depend on the covariance that we've observed in the historical time series. And so if the covariance changes, like if one CU becomes more vulnerable because of change in migration or a change in the fishing practices, then that's not captured in these models. And so then we need to be more precaution. Then we need to kind of rejig things. One of the benefits of this projection-based LRP is that we can kind of add in some hypothetical or some plausible parameters or covariance structures. If we think that the covariance is actually different now than it was historically, we can put that into the projection model, say, if the covariance has changed in this way, how is that going to affect the probability of any single CU being below the lower benchmark, and identify an LRP associated with that. So you can kind of, with the projection, you kind of play around with what the vulnerabilities or the covariances are a little bit.
Okay. So let me play devil's advocate. Isn't part of the problem the fact that our target harvest rates are potentially too high, that we don't know what kind of impact we're having on the system as a whole, and so by treating these stocks more or less in isolation from the rest of the environment, the reality is that we may be impacting our ecosystem, changing the productivity of the system in some way, and simply leaving more fish in the water would be a better strategy than the approach that's currently being taken.
Yeah.
I had to throw that at you.
Yeah, no, definitely. But that's like leaving more fish in the water. It's kind of like saying having a higher LRP. And if we don't, like if the stocks aren't covarying very well, then that's what these methods will do is they'll give you a very high LRP, which makes you leave more fish in the water. So I think that captures exactly, but the challenge is you have to be able to parameterize how the stocks are covarying. And a lot of the cases, like for salmon, we do have some cases where we understand what the CU level population dynamics are. But I think in a lot of these, if we apply this to other systems, we know that there's population structure, but we don't know how they vary. We don't know how weak the weakest stock is. And so that's the challenge then, because we don't know how many fish we need to leave in the water in order to protect that weakest stock. Yeah.
Okay,
So yeah,
thanks.
it's a challenge. And for salmon, perhaps we have slightly more data than we would for other systems where the population structure, where we just don't have as much information at the finer level to kind of make those decisions.
I would say that for anything that isn't salmon, we probably don't have that knowledge.
Yeah, probably. Yeah. Yeah, great questions.
Right. So go ahead and raise your hand if you have a question. I'll call on you. In the meantime, I did have a question about splitting up this data for a time varying model, and what kind of data demand does that take year to year, and is it feasible to get that data every single year for all these little management units?
Yeah, great question. We've applied this to Fraser Sockeye, which is our data rich system. So we do have annual estimates for all of our stocks. When you have, so in our time series range from probably 30 years to 70. Yeah, as your time series gets shorter, like less than maybe 15, it gets harder to identifying the underlying changes in productivity, because these models are looking for kind of that they're separating the interannual noise from the underlying trend. And so if there's data, if you're less than 10 years, then the model can't really separate that interannual variability from an underlying persistent change. So there are data demands. Yeah. There is work to identify kind of meta population versions of these time varying models where you can harness information from multiple stocks in a hierarchical context to get at what are common underlying patterns. So that reduces the data demands on each individual stock when you're analyzing them together. There was a recent paper by Tableau, I think, on the East Coast that looked at this. Yeah.
Oh,
So
cool,
I
thank
guess
you.
the methods are evolving is what I'm saying.
Yeah, I see.
Yeah, and in really neat
Yeah,
ways.
thanks.
Yeah, and for our stocks, we do have, like the stocks that I showed, these are the data-rich examples.
Yeah, yeah,
Yeah,
it'd be interesting to compare that, like a time varying model compared to maybe in an area that's recently had some development compared to an area that might not have had any, and see,
yeah,
how much reference points changes just with development or anything like that.
yeah. Yeah, and so we're hoping to do this in our work right now is to document evidence for changes in productivity and capacity, across the coast and relate that to kind of environmental conditions and change what we know about changes in habitat, changes in like forestation and other like threats, and how that might kind of impact our ability to detect those time varying changes and their impacts on the reference points.
Yeah, cool. That's exciting.
Yeah. Yeah, and then as Pierre knows, there's DFO is has a requirement now to more fully consider kind of ecosystem and environmental considerations in our advice. And so it's becoming more, there's more pressure on us as we provide our advice through reference points that we are considering, what are the recent environmental conditions and how are these changing population parameters and threats impacting this advice that we're providing.
Yeah, cool. That makes sense. That's good that there's an initiative for that. I know when we had Dr. Billy Christensen give a talk last semester, he was like, "Come on, ecosystem stuff." Obviously ecosystem, but or ecosym and that stuff. But yeah, he was really harping on getting more ecosystem stuff involved for sure.
Yeah, yeah. I think it'd be great to have Pierre give a talk about how DFO is doing this in general.
Yeah. All right. Well, I think that's it for questions. Yeah, before I end it though, I do want to thank you, Dr. Holt, for being here with us, and that was an awesome talk. I really appreciate that. I do want to introduce next week's seminar. Before we leave, though, we have local bird legend and seabird behavioral ecologist, Dr. Bill Montevecchi, who's going to be talking. His seminar is titled "Seabird Risk Assessment: Interrogating Ocean Ecosystem Change." All right. So yeah, with that, I'll wrap it up, I guess. And everybody have a great Wednesday.
Thanks, everyone. Thanks, Carrie.
Thanks.
Bye.